let's start with Chapter 9 today. We're talking about the cell cycle and cell reproduction. Some of the questions that we're going to answer today are, how do cells divide and why they divide, when they divide? I'm sure these are burning questions that keep you up at night. Yes. Some of the things we're going to talk about is how in the world does a cell divide and get all the right stuff into two cells? We're going to talk about how often they divide, and is division the same for all cells? We've talked about lots and lots of different types of cells this semester. We've talked about prokaryotic cells, eukaryotic cells, plant cells, animal cells. Is it the same for all of them? Also, we're going to end the chapter with what happens when cell division goes wrong. We've got a short segment on cancer and how cancer is associated with cell division. First of all, let's go back and review for a moment about unicellular organisms or single-celled organisms. When we talk about division in single-celled organisms, we talked a little bit about this in Chapter 4, and we've got two chapters toward the end of the semester, a chapter on protists and a chapter on bacteria. So we're really not going to spend a whole lot of time in Chapter 9 talking about single-celled organism division. But one thing we have mentioned is that when a single-cell organism divides, that's actually how they reproduce. So cell division in single-celled organisms is called asexual reproduction. And if you'll recall, one of the things that we talked about was that when an individual cell divides to produce two cells, both of those cells would be genetically identical to the original. Okay, so there's no genetic variation when you have asexual reproduction. Okay, a little bit of terminology as we go forward, just so that you won't be confused, because you're going to see this in some of the literature that you look at. When a cell divides to produce two cells, regardless of what type of cell it is, oftentimes in science and biology, we refer to the cell that goes into division as the mother cell, and the cells that come out of that division as the daughter cells. Okay? It has nothing to do with gender or reproduction or anything like that. It could have just as well been father cell and son cells. Okay? But I just don't want you to get confused when we start talking about daughter cells. When I say the daughter cells of a division, I'm merely referring to the two cells that resulted from that division. Does that make sense? Make sense? Okay. So a lot of students get real confused when we start talking about mother cell and daughter cell, and how does that relate to reproduction? It doesn't. Those are just terms that we use, terminology that scientists use to talk about a cell going into division and that cell producing two cells. Okay, mother cell and daughter cells. Okay, so in asexual reproduction, for a single-celled organism, we really could say that division is how they multiply. Catchy little phrase there. Division is how they multiply. So let's talk about multicellular organisms. The rest of the time will be on multicellular organisms. For instance, you. Okay, obviously you are a multicellular organism. When do your cells divide? When do the cells of your body divide? They divide for growth or repair. Your cells, the cells of your body, divide for growth or repair. You're either growing, and growing is merely producing more cells, becoming more, having more mass. Repair would be when there's death of other cells. Anybody know who that is? Growth, that's exactly right. Looks pretty bad, doesn't it? I know, it's hard to look at. Growth and repair. So when do your cells divide? For what two reasons? You're either growing or you're repairing. So right now in your body, you have cells that are constantly dying and need to be replaced. So you still have cell division occurring in your body as those cells are being replaced by new cells. So your cells divide for growth and repair. So what does it function to do? What does this division do? It actually maintains that surface area to volume ratio. If you'll recall from chapter 4, we talked about how cells have to stay small because the surface area is the plasma membrane and the volume is the content of the cell that needs to be have nutrients supplied, and oxygen needs to have carbon dioxide removed and other waste products. And the cell has to be small because as a geometric figure, like the one that we're looking at here, increases in size, the surface area and volume do not increase at the same pace. The volume increases faster than the surface area. And at some point in time, the plasma membrane would not be able to provide the nutrients for the whole content of the cell. Another thing that it does is it passes a complete set of chromosomes to every cell. Cell division functions to transfer a complete set of chromosomes to every cell. I think it's amazing to think about that at some point in time in the past, you were but yet a sperm and an egg that had not fused yet. And during that moment of conception, when that sperm and that egg fused, and I know you don't like to think about that, because that means your parents did it at least once, okay? But when they fuse, that egg and that sperm contributed that genetic information to make you who you are. Those 46 chromosomes that were present at that moment, those exact chromosomes were copied into 2 cells, and then those 2 cells copied it into 4 cells, and then those 4 cells copied it into 8 cells, and then after millions of divisions, we get you. Okay, which is amazing to me that every single cell in your body has a copy of those original 46 chromosomes that were in that zygote the moment that you were formed. Let's do a little review. If you'll recall from chapter 4, we talked about the nucleus and the chromatin in the nucleus, and we talked about the fact that when your cell's not dividing, your DNA actually exists as these long, stringy strands of chromatin that are really, really diffuse, and you can't see them. You can't, even with our light microscopes, you couldn't see chromatin. But if we look at it and take the chromatin into an electron microscope and unravel it here, we would eventually be able to see the double helix structure right there, which is your DNA. I love it that most textbooks show chromatin that looks like a bowl of spaghetti. Again, if we can relate it to a food item, I can remember it. So there's your chromatin, and we unravel it, we look down there, and there's your DNA. Again, this is in a non-dividing cell. So here's my DNA. It wraps around all of those proteins that you see right there. So this would be my chromatin in this illustration right there. And if you'll recall, we said that before cell division even occurs, your 46 strands of chromatin make copies of themselves. They're preparing, your cell is preparing to divide, and it knows that every cell has to have 46 strands of chromatin. So even before it goes into division, it's preparing, and it copies every piece of chromatin, and it holds on to that piece of chromatin, that copy, by that structure right there, which we're going to call a centromere, and we'll get that in your notes in just a moment. So this is replicated chromatin. So going into cell division, it's a whole lot easier. It's a whole lot easier to move things when they're compacted. And you told me that when we talked about this in chapter 4. I asked you, what do you do before you move to a new apartment? And you said, I pack up my stuff. So this is exactly what the cell's doing. Right before cell division, it takes those long, stringy strands of chromatin and it condenses them into what we're going to call chromosomes. Okay, so there's your chromosome right there. It is a replicated, condensed piece of chromatin. Some books even refer to this structure right here as a duplicated chromosome, just emphasizing the fact that both of those are exactly identical to each other. Every species has a certain number of chromosomes. How many did I say you have? 46. Okay, you have 46 chromosomes. And when I first was studying genetics, and somebody said you have 46 chromosomes, I thought, well, gosh, that must be the most of any organism out there because we obviously are the most highly evolved, right? So I thought 46 chromosomes is going to be the most anything has until I saw this little chart right here. And it says that chickens have 78. Okay, so chickens have more chromosomes than we do. Take a look at cats. Cats have 38. They even have more chromosomes. As a matter of fact, potatoes even have more chromosomes than we do. So what's that all about? Obviously, it's not the number of chromosomes you have. It's what's coded on those chromosomes, right? I mean, take a look down there. If the number was what was important, then dogs and chickens would be identical. Look at that. Dogs and chickens both have 78 chromosomes. So obviously it's not the number. It's what's contained on those chromosomes. But take a look at them. What do you see? What do you see that all of these numbers right here have in common? They're all even. That's exactly right. It's because chromosomes come in pairs that look alike. And we're going to call them homologous pairs. Chromosomes come in pairs that look alike, and we call them homologous. So here's an illustration, a picture, an actual picture of human chromosomes right there. Notice that there are 23 pairs, 46 chromosomes, and we call the picture of a set of chromosomes a karyotype. Now, when I say that chromosomes look alike, or they are homologous, and remember that homo means the same, that means that they're the same size. They look alike, they're the same size, and that connecting area right there that's holding the copies together that we call the centromere on a pair of homologous chromosomes will be in the same location. So for this particular pair of chromosomes, there's the centromere. For this pair down here, the centromere is right there. Notice that the centromere is right there on that little pair. So if they look alike, they're the same size. They have the same centromere location. And if you were to stain these chromosomes, the pairs would stain alike. When you stain a chromosome, and I've got lots of illustrations later on to show you, but when you stain a chromosome, it appears striped. So when the chromosomes are homologous, the stripes are exactly the same. Okay, so that's what I mean when I say they look alike. Same size, same centromere location, and they have the same striped pattern or banding pattern as we call it. Now, keep in mind that chromosomes in a cell do not hang out with their homologue, or the one that they are homologous with. If you were to find a cell where you could see the chromosomes, and again, it would have to be a cell that was very close to dividing or in division, because that's the only time that you see them condensed like this. It would have to be close to division or in division to be able to see these. Okay, so when you look at a nucleus that you can see the chromosomes, they're going to all be jumbled up together. And what the geneticist does to make the karyotype is he cuts out the chromosomes. Literally, a long time ago, they used to have to take a picture of them, a photograph, and cut them out and then paste them together, literally. Now we have all kinds of technology that allows you to do that with a computer. But they cut the chromosomes out that are homologous to each other, that look alike, and they would line them up like you see here in this format. Okay? And then they make sure that the largest one is number 1 and the smallest one is number 22. So they line them up based on size. The 23rd pair of chromosomes, reserved for this spot right here in the bottom, is what's called your sex chromosomes. These chromosomes determine your gender. And you see that this individual, and this is a real eukaryotype, this individual is a female because she has two X chromosomes. Had this individual been a male, there would have been one X chromosome here and then a Y chromosome here that is very small. The Y doesn't look anything like the X, so they're not homologous. Okay, the two X's are homologous there. But notice how they're lined up, the first 22 pairs by size, and then the 23rd pair is the sex chromosome. Okay, and keep in mind that every human being's chromosome number 1 would be that exact same size. Your chromosome number 1 is going to look just like my chromosome number 1. Okay? Your chromosome number 2 is going to look just like my chromosome number 2. Now look, did I say they're identical? No. I said they look alike. That means they're the same size, they have the same centromere location, and they would have the same banding pattern. Okay? So this is what's in your cells, your body cells. Okay? 23 pairs of chromosomes. We call this condition diploid. Di means 2. So that literally means there's 2 of each kind. There's 2 number 1s, there's 2 number 2s, there's 2 number 3s, 2 number 4s, and so forth and so on. Okay, now, if this is a cell in your body, and there's 46 chromosomes, where did these come from? They came from a sperm and an egg, right? These 46 chromosomes came from a sperm and an egg. So half of those chromosomes were from your dad. Half of these chromosomes were from your mom. Not just any half, it's going to be half of each pair. So of that pair number 1 of those was in your dad's sperm and one of those was in your mom's egg, as was all of these other pairs. One comes from the mother, one comes from the father. Okay? So the sperm and the egg had 23 chromosomes in it, right? So when the 2 came together, it restores this condition that we call diploid that has 46 for humans. The egg and the sperm we refer to as haploid. They have half. They have 1 copy. Haploid is in. Diploid is 2. 2 in. Okay, if you'll recall, when we were in chapter 4, we talked about how a cell starts out small and it grows and it grows and it grows, and then it gets so big that that surface area to volume ratio is getting way, way too small, and the cell has 2 choices. It can either die, because it's eventually not going to be able to bring in enough nutrients and oxygen to keep the cell alive, or it can do what? It can either die or divide and become 2 small cells. Those events that occur from division, from coming out of division until that cell divides again, we call the cell cycle. It's everything that occurs in that cell between divisions. The events in a eukaryotic cell between one cell division and the next. That is called the cell cycle. So the small cell grows and grows and grows and grows, gets big, it divides into 2 smaller cells. That would be one cell cycle. Okay, the 2 small cells start to grow, 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 grow. They get big, they divide, it starts over. Okay, we're going to take those events and we're going to divide them into 2 stages. We're going to call it interphase and the mitotic stage. Inter means what? What is inter? Between. Okay, so interphase would be all those things that are happening after division until the next one. Okay, so that's what's happening between divisions. The mitotic stage would be the dividing. Interphase is divided into G1, S, and G2. The M stage, which we call the mitotic stage, which we sometimes call the M stage, is divided into mitosis and cytokinesis. Okay, so interphase is divided into G1S and G2. The mitotic stage is divided into mitosis and cytokinesis. So, cell starts out small, it goes straight into G1, then to S, then to G2, and then into M, where it divides. The resulting cells, or the daughter cells, would go right back into G1. So it's a cycle. It's the cell cycle. It goes in a circle. So let's just look at it as a circle right here. The blue that you see here, that is interphase. There's your G1, there's your S, there's your G2. The green portion is the mitotic phase, which would be mitosis right there and cytokinesis. So that's your M stage. Mitosis and cytokinesis are two separate events. In most cells, they occur simultaneously, but they are separate. So you've got a cell that comes out of division. It goes into G1, S, G2, and M. The cell divides into two cells. Those two cells go straight into G1. So it's a cycle. It's a continuum. It goes on and on and on. So let's just talk about what happens in each one of these. It's a circle, so it's hard to know where to begin. Does the circle have a beginning? So we'll just start with G1. When G1 occurs, where did the cell come from? The cell just came out of division. A cell in G1 just came out of division. So it's going to be really, really small. So the first order of business, the first order of business in G1 is for the cell to grow. It's very small. It needs to grow. It got half of the organelles of the original cell, the mother cell, so it needs to produce more organelles. The cell then progresses into the S stage. S stands for synthesis. This is where DNA replicates itself. This is where chromatin makes a copy. That's what we're talking about when we say chromatin replicates. We're talking about making another molecule of DNA. The cell then progresses into G2. G2 is right up next to division, so the cell's beginning to prepare to divide. To be able to divide and tear those chromosomes apart and put them in new cells, the cell has to have proteins. Division involves movement. Anything that involves movement is going to use proteins. So proteins are being made so that those chromosomes can be torn in half. The cell continues to grow as this occurs. So that is interphase, G1, S, and G2. We've now reached a time in the cell cycle where the cell has to divide. So we go into M, which we said was mitosis and cytokinesis. Mitosis is defined as nuclear division. Cytokinesis is division of the cytoplasm into two separate cells. So the result of mitosis is two nuclei. The result of cytokinesis is two separate cells. Okay? So what would happen, let me just ask you this, what would happen if I have a cell that undergoes mitosis? So what does it have? Two nuclei, but it doesn't undergo cytokinesis. What would you have? You would have a single cell with two nuclei, and that happens. You have some cells in your body that are multinucleated. That means that they have mitosis without cytokinesis. There. The whole cycle, the whole progression from G1 to S to G2 and on to M is controlled by a myriad, lots of proteins. A major component, a major part of that are proteins called cyclin, C-Y-C-L-I-N. These are internal signal proteins that tell the cell when it's okay to go from one stage to another. And how this works is the amount of cyclin in the cell will go up and down. It increases and decreases as the cell cycle continues and progresses, and the cell interprets that amount of cyclin as an indicator to either stop or start or pause. Okay, so cyclin will increase and decrease in the cell as an indicator to the cell that everything's either okay to progress or no, you need to stop. You need to pause and see if you can fix the mistakes. Okay, so cyclin will go up and down. So what happens is there are several very, very important checkpoints within the cell cycle where if cyclin signals and the cell pauses and the cell checks to make sure things are right. And if it's not right, then the cell will stop. The cell cycle will not progress. From G2 to M is very important, and from G1 to S is very important. These are checkpoints in the cell cycle. Let's take a look at where those are and think about what's going on. Right here, here's my cell cycle, G1, S, G2, and M. We have a checkpoint right here from G1 to S. Think about what's happened to your genetic information at this point. During the M stage, right here, you had a chromosome that was literally torn in half and put into two cells. If you're a human, you had 46 of those. 46 chromosomes torn in half and put into two separate cells. Those two separate cells start growing in G1, and it reaches right here. What's going to happen to those pieces of chromatin during the S stage? They're going to be duplicated. That's right. Well, before you duplicate, wouldn't you like to check to make sure that those were torn apart correctly during division? So after you tear them apart here, you check to make sure that everything went okay, everything's as it should be, before you make a copy of it. Because if you make a copy and something's not right, what do you end up with? You end up with two copies of something that's not right? So you want to make sure everything's okay before you copy your chromatin. So after this checkpoint, chromatin is copied. We're still diffuse. We're going into G2. What's going to happen to my chromatin right here? It's going to be torn apart and put into two cells, right? Wouldn't you like to make sure copying went okay before you separate them? Because if something happens here to your DNA and you don't check it, then you've got the chance of putting something that's mutated into a cell by itself, and it goes off and starts dividing and producing more cells that have mutations in them. Okay, so you check it after you copy it, before you divide it to make sure everything's okay, and you check it after it's divided before you copy it. Okay? And again, if something goes wrong and it's not right, the cell pauses and hopefully gives the cell time to correct the mistake. If the mistake can be corrected, the cycle continues. If the mistake cannot be corrected, the cell is programmed to self-destruct. It's called apoptosis. We talked about this a little bit when we talked about lysosomes. Apoptosis is programmed cell death. There are enzymes within the cell contained within lysosomes that are called caspases that are required for apoptosis to take place. These things are normally held back, but when the cell senses or realizes that something's wrong that it can't fix, these caspases are unleashed and the cell literally digests itself. So you should be asking me a really important question now when I just told you that mutated cells, cells that are genetically inferior, destroy themselves. Why do we have cells that are mutated then? Which is basically what cancer is. Cancer is the cell cycle out of control. We'll talk about that more in just a moment. But why, if the cell is programmed to destruct, if something can't be fixed, why do we have cells that go bad? Has anybody got a clue? I want you to think about it for just a moment. We said we have all kinds of proteins, enzymes, that are trying to keep our cells healthy, trying to monitor. We have things called proof checkers that constantly scan your DNA up and down and make sure that A's are with T's and T's are with A's and C's are with G's and everything's right. But all of these molecules involved in keeping your cells healthy are proteins. Okay, well, you think about that for a moment because you already know from chapter 3 the code, the recipe to make a protein is contained within DNA, right? So these enzymes that are keeping your cell healthy, the recipe for the cell to make them is contained within the very thing they're monitoring. It's kind of a catch-22. If you get a mutation within the recipe or the gene that codes for one of these really important molecules, then you don't have that anymore. Your cells can't keep themselves healthy if they don't have the recipes to make the molecules to do that. Okay, there's our bowl of spaghetti again. Remember, before nuclear division, this chromatin will replicate and condense and will look like this, literally. That is a picture of a human chromosome. Some books, as I mentioned earlier, call it a duplicated chromosome. But let's get some vocabulary down so we can start using the right words. The thing that holds the two copies together is called a centromere. C-E-N-T-R-O-M-E-R-E, centromere. Keep in mind that one of these used to be the single strand of chromatin. It replicated itself, so this entire structure right there is identical, exactly the same as this structure on this side. Each one of them is called a chromatid. So this side by itself is called a chromatid. Together, remember they're identical, we call them sister chromatids. Sister implies that they are identical. And again, I don't know why we call them sister chromatids instead of brother chromatids. I don't know. It has nothing to do with gender. Okay? Sister chromatids just means that they are identical to each other. You guys remember these structures right here? The ones that look like Twizzlers? You remember those? Those were what? Centrioles. That's exactly right. We said animal cells have them and plants don't. Well, these centrioles that we said we've already defined, they're involved in cell division. These centrioles come from a place called the centrosome. So those are our centrioles. And they are responsible for producing what's called the mitotic spindle that will produce spindle fibers. Okay, there are fibers, there are protein fibers that are produced that are responsible for moving our chromosomes. We have to have a way to move them around, and these things are going to produce the proteins that do that for us, the mitotic spindle or spindle fibers. Okay, so let's just take a look at the whole process here. In an average cell cycle of 24 hours. I love this illustration because it takes a cell cycle and puts it in an amount of time that we actually really can relate with. Some cells divide longer than 24 hours. Some cells divide shorter than 24 hours. 24 is just an average length. Some cells don't ever divide at all. They're arrested at a certain stage and they don't continue to divide. But on a 24-hour cell cycle, take a look at this. If this entire cycle is taking 24 hours, which stage is the cell in the most, the longest period of time? G1. Okay, what's it doing during G1? It's growing and increasing the number of organelles. The next longest would be the S stage, where DNA replication, we're copying our chromosome right there. And then take a look at the shortest one in interphase, it's G2. So once DNA is replicated, then it's time to go straight into division, pretty much. But the one thing I wanted to really emphasize with this illustration is the length of time the cell spends in the M stage. What do you think that is? About what? 2/12? 1/12? Okay, so maybe about 1/12 of the cell cycle is spent in mitosis. In a 24-hour period, that would be a couple of hours. I think this is pretty eye-opening for most high school students because having taught high school in the past, I know how much we emphasize mitosis. We talk about mitosis all the time. And I think students lose sight of the fact that cells don't live to divide. Division is a result of growth, and cells are only going to divide when they reach a size that they have to, or if we have an injury, okay? But take a look at this, 1/12 of the cell cycle in division. So what is the cell doing for the other 22 hours? It's being a cell. I mean, think about it. We're talking about liver cells, muscle cells, kidney cells, skin cells. These cells have a job to do. So during G1S and G2, if it's a liver cell, it's doing what liver cells are supposed to do, okay? And it only divides when it has to. I just want to make sure you realize that we talk so much about mitosis, and it's very important, but that's not the ultimate goal of cells, okay? The cells have jobs to do, and division is a result of growth. It divides when it has to. Okay, so let's talk about the M stage, cytokinesis and mitosis. Mitosis, again, is nuclear division. Cytokinesis is division of the cytoplasm. So let's go straight into mitosis. We're going to divide mitosis, the events of mitosis, into 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase. The 5 stages of mitosis. Okay, so let's start with prophase. First of all, before we get started, let's make sure we understand what we're looking at here. We're going to use this example for most of the Chapter 9 and Chapter 10. They use this exact example of this cell. The diploid number for this organism that we're looking at here is 4. There are 4 chromosomes. Notice that there are 2 long ones, one red and one blue, and 2 short ones, one red and one blue. So if I were to show you this, which pairs would be homologous? Of these chromosomes, which are homologous? The big ones are homologous and the little ones are homologous. Chromosomes are not normally pink and blue, okay? So if they were all the same color and I said which ones look alike, you would have naturally said the long ones are alike and the short ones are alike, right? But notice that there is a red long one and a red short one, and a blue long one and a blue short one. They are red and blue for a reason. What does that mean to us? That means that for every pair, the blue and the red, one of them came from the mother and one came from the father. So the sperm for this organism would have had those 2 chromosomes right there in it, and the egg would have had those 2. Okay, so that's why they're red and blue. So what happens in prophase? Well, to begin with, we already had duplicated condensed chromosomes. So our chromatin has replicated and condensed. We already have condensed chromosomes. And notice how our centrioles right here are starting to produce these yellow things. This would be the mitotic spindle right here. We're starting to produce spindle fibers. So our centrioles are producing the spindle fibers. At the same time, notice how the centrioles are starting to migrate or move to opposite sides of the cell. This centriole will end up on the opposite side of the cell as this centriole as they produce these spindle fibers. The ultimate goal of this is for those spindle fibers to attach to our chromosomes, pull them apart, and put half of each chromosome into a cell. Okay? For all, if it's a human cell, it'll be all 46 of them. For this, it'll be all 4. So to do that, we have to get rid of that barrier, the nuclear envelope, that's going to keep those spindle fibers out of the nucleus. We got to get rid of the nucleus, basically. So our nucleolus disappears and our nuclear envelope disappears. So we've gotten rid of the barrier to get into the nucleus. The next stage, and keep in mind that as we go through these stages, the cell doesn't do prophase and then stop and say, well, I think I'll do prometaphase and then I'll do metaphase. And it doesn't do that. This is more of a continuum. And we say the cell has progressed into prometaphase when certain events have happened. Okay, so it's a flow of events. The next is prometaphase. What do you see that's different here? What do you see? Those fibers, the spindle fibers, have attached to the chromosomes. That's exactly right. Our centrioles are now on opposite sides of the cell. The centrioles have spindle fibers that have attached to the chromosomes, and there's going to be a spindle fiber from each centriole attached to each chromosome. After they attach, then these spindle fibers pull and push. It's almost like bungee cords. They pull and they push, and they're going to line up and move these chromosomes into the middle of the cell. So we say that the cell has progressed to prometaphase when the kinetochores develop on the centromere. The kinetochores are what the spindle fibers are going to attach to, and the spindle fibers have attached. So these kinetochores are what attach the spindle fiber to the chromosome. So if I were to ask you, spindle fibers just attached to the chromosomes, what stage am I in? You would say prometaphase. Now that's the benchmark. We've moved into prometaphase when those fibers attach to the chromosome at the kinetochore. Once they attach, now we can start moving our chromosomes around. They're going to move the chromosomes to the middle of the cell, at which time we call that metaphase. Metaphase is when the chromosomes are lined up in the middle of the cell. Anytime you see the word meta, think middle. Metaphase means it's in the middle of the cell. I mean, this is pretty easy. Look at this, guys. We've got them all lined up right here. Now all the cell has to do is tear them apart and pull each of the half to its side of the cell, and then you end up with 2 different nuclei. So we're going to tear these apart. This part right here is going to go over here, and this portion right here is going to come this way. And we call that anaphase. That's when those sister chromatids split and each half is pulled to an opposite pole. Once the single pieces of chromatin, once it becomes diffuse, it'll be chromatin, but once the single-stranded chromosomes make it to the other side of the cell, we say the cell has progressed to telophase. I want you to think of telophase as the opposite of prophase. Okay, everything that happened in prophase is being reversed. We're forming two new nuclei here. So in prophase, we said that the spindle fibers formed, right? Well, in telophase, they disappear. In prophase, we said the nuclear envelope disintegrates. Well, in telophase, we're going to form two new nuclear envelopes around our two new nuclei. In prophase, we said that the chromosomes condensed. Well, in telophase, those single strands are going to become chromatin. And in prophase, we said the nucleolus disappeared. Well, it comes back in telophase. So telophase is really just the opposite of prophase, but you're doing it. It's happening to two new nuclei. Notice this right here. This is called the cleavage furrow. Does everybody know what a furrow is? Have you ever furrowed a garden or furrowed a field? That's where you make that little trench that you can, you plant your seeds in. So this is a furrow. This is how cytokinesis starts in animal cells. Cytokinesis, we said, was division of the cytoplasm. And again, it does accompany mitosis in most cells. It begins in anaphase. It will continue through telophase, and it ends at interphase. And it starts with that cleavage furrow right there. And remember, we said that if a cell experiences mitosis without cytokinesis, you have a multi-nucleated cell. Your striated muscle cells are multi-nucleated. So we're going to start with this cleavage furrow right there in cytokinesis. We start with a cleavage furrow, and then we get the formation of this structure right here called the contractile ring, which literally squeezes the cell in two. We start with a cleavage furrow. The contractile ring forms between these two daughter cells, and it's going to squeeze the two cells in half. It's going to squeeze them in two. Have you ever taken a ball of Play-Doh in your palm and squeezed it so that the Play-Doh comes out both sides? That's exactly what happens. So this will eventually look like that. There. It's being squeezed in two. Mitosis in plant cells is very similar. In plants, if you've ever planted a garden, you know that from where do plants grow? If you're watching a plant grow, where do you see the new growth? Where do you see it? At the shoot, okay? And they also grow from the roots, okay? So the new growth of plants occurs at the shoots or the roots. These tissues, the tissues located there, are called meristematic tissues. And that's where a plant grows. And those tissues, the cells of those tissues will undergo mitosis throughout the life's plants as it produces new growth. So meristematic tissue is where a plant grows from where it grows. They're found at the root and the shoot. And mitosis for a plant looks very similar to mitosis for an animal. This is a stained cell. Obviously, it's stained, but it is a plant cell. This is prophase. You see we have replicated condensed chromosomes there. In metaphase, they're lined up in the middle. In anaphase, they're pulling apart. And in telophase, we're having two new cells being formed here. So mitosis in plant cells is very similar to mitosis in animal cells. One of the primary differences is cytokinesis. Plant cells do not furrow because of the cell wall. Remember, they've got the rigid cellulose cell wall around them that will not allow for furrowing. So what you have is the formation of a cell plate. These vesicles here are lining up. The vesicles contain the components to lay down the cell wall. for those two cells. So you have these vesicles lining up. That's the cell plate that will eventually be the cell wall for those two cells right there. And here's a photograph of an actual plant's cell plate right there. So cytokinesis in plant cells, very different than cytokinesis in animal cells. No further. With regard to prokaryotic organisms, we can't call cell division or in prokaryotic organisms mitosis. We can't refer to any part of it as mitosis. Why? What is mitosis? Mitosis is division of the nucleus. What is a prokaryotic cell? It's a cell that doesn't have a nucleus. That's exactly right. So we have to call it binary fission. Binary fission literally means splitting in two. Binary means two, fission is to split. So we're talking about a prokaryotic cell splitting in two. They don't have a nucleus. They also don't have a lot of those other membranous organelles that we talked about in eukaryotic cells. They don't have mitochondria. They don't have Golgi bodies. They don't have any of the membranous organelles. They do have a single chromosome. It is circular, and remember from chapter 4 again, it's in an area called the nucleoid. Okay, remember we talked about those three words that were very confusing: nucleus, nucleolus, and nucleoid. The prokaryotic chromosome is in the area of the cell called the nucleoid. So let's take a look at what binary fission looks like in a prokaryotic cell. If this is my prokaryotic cell, this purple structure is my single chromosome, and it's circular. Before division starts, the chromosome attaches to the inside of the plasma membrane right there. It's about to divide, so it attaches. After attachment, the chromosome will replicate itself. So in this illustration right here, we have two of the circular chromosomes right there. The cell then starts to elongate or pull. As the cell elongates, those copies will be separated because they're still attached to the plasma membrane. The cell continues to elongate here. Cytoplasm is distributed evenly between the two cells. You have some cleavage starting right there, and then the cells will split in half, with each of the cells having a copy of the single chromosome there. So it's very simple compared to eukaryotic cell division. But then again, prokaryotic cells are more simple than eukaryotic cells. Okay, so what happens when cell division goes wrong? Okay, again, the cell cycle is controlled by many molecules that are primarily proteins. There are lots of cases where those proteins are not functional that will result in cancer, that we define as a growth disorder that results from the mutation of genes that control the cell cycle. Now that's a long sentence. Let's think about what that just said. A growth disorder. So the cell cycle is out of control. Growth disorder. Cell cycle out of control. That results from mutation of genes. That's the recipe right there. Mutation, the recipe's changed in the proteins that regulate the cell cycle. Okay, one of the reasons cancer is so hard to combat is because your body doesn't recognize it as a foreign invader. These are your own cells that are out of control. So your body doesn't see it as a threat. It's not a virus. It's not a bacteria. Okay, now there are some viruses that can go in and mutate your DNA and cause those proteins not to be functional, but the virus isn't causing the cancer. Okay, it's the gene being mutated that causes the cancer. Before we go on, I want just a moment. Everybody that's been touched by cancer, At some point in your life, you either someone you love got cancer or you lost somebody to cancer, but you've been touched by cancer personally in your life. I want you to stand up. Stand up. And guys, I want y'all to look around at this. Okay. Okay. The development of cancer is called carcinogenesis. And that's the development of cancer, and it tends to be gradual. And I'll explain what I mean by that in just a moment. But the development of cancer tends to be gradual. And most of you know that a substance that causes mutations of your DNA is called, that would cause cancer, is called a carcinogen. Okay? Okay, when I say that the development of cancer tends to be gradual, let's talk about that for just a moment, because that's sometimes hard to understand. Keeping in mind that cancer is a result of mutations in genes that code for molecules that control this whole cycle. Okay, it's basically a cell out of control. When let's say you go and put a cell in a copying machine and you put, I want 2 copies and press copy and out comes 2 cells, right? Well, cancer is like pressing 2 copies and pushing go and the copying machine gets stuck. And you don't get 2 cells, you get 2 million. Okay, it's just copy, copy, copy. It's out of control, it's stuck. And it just continues to produce cells. Okay. Well, let's go back to your DNA for just a moment. And let's use an example. Do y'all know what a continuum is from one point to another? Let's say that right here, from here to here, okay, right here at this point would be the perfect DNA sequence to produce a protein that's involved in the cell cycle. Okay, right here is the perfect sequence. This is how it should be. It's producing the perfect protein. Everything's good. And go back to chapter 3 and think about what a protein is and how they work. What's important about a protein is its shape, because they literally fit into other molecules, and that's what makes things happen. Okay? So here is the perfect protein. Let's say, for instance, that you're born with the perfect sequence. Well, every time a cell divides, every single time a cell divides, mistakes happen. Now, your proof checkers catch most of them and fix them. But every time your cells divide, some of those mistakes slip through. And that's what a mutation is. Now, guys, a mutation doesn't mean that you're going to end up with 2 heads and 6 fingers and, 4 toes or whatever. The definition biologically of a mutation is a change in the genetic code. Now, whether or not that results in a change in the phenotype, it depends on what that is. You don't know what phenotype is yet, I'm sorry. Whether or not that results in a change in your body, okay, it depends on what the mutation is. But a mutation is just a change in a code. Maybe a T is not a T anymore, it's a G or a C or an A, okay? So every time your cells divide, some changes slip through. So you get an accumulation of changes over time. And the word accumulation is the important word here. Okay? Every time a cell divides, changes occur. And then they divide again and changes occur. And they divide again and a change occurs. Well, some of those small changes might not affect the shape of the protein, but they're cumulative. Okay, let me use another example. Let's say it's a recipe. It's a real recipe. Those of you that like to cook. Let's say over here we've got a recipe for a pound cake. No, let's say what's your favorite kind of cake? Let's say red velvet. I love red velvet. Y'all like red velvet? Okay, so we've got red velvet and cream cheese icing. Okay, oh my gosh, I'm starting to drool. Perfect recipe. Okay, it calls for down swan cake flour. Y'all know what that is? That's really expensive cake flour that makes like the best cakes ever. Okay, so we got this recipe, it's perfect. Well, then we decide to change it a little bit. So instead of cake flour, we're going to use Great Value self-rising. Okay, so it's not cake flour. Okay, definitely not going to be the same thing, but it is a cake and it's similar, right? So let's say we make it, we've changed that, we're going to make another change. Instead of having the red velvet center, we're just going to make a plain old white cake. Okay? So it's still a cake. We've made changes to the recipe. It's still a cake, but it's not the same at all, is it? And then all of a sudden we make another change. Let's say we decide to put a fourth of a cup of sugar instead of 2 cups of sugar. Okay? It's still going to be a cake, right? Is it going to be the same? No. And then the next change we decide to leave out the eggs. What happens? It's not a cake anymore, is it? Okay? You got to think about DNA and proteins the same way. You could start out with the perfect recipe, and as you live, guys, just as your cells divide, as you are exposed to toxins, okay, substances that cause genetic change, you get additional accumulated changes. And then all of a sudden, all of those changes together, you're on the edge of a cliff, and all it took was one more change, and you fell off the cliff. That cell developed cancer. Okay? And the whole problem here is that you don't know where you are in this continuum. You're not born with the perfect sequence. Okay? You could be anywhere along that continuum from perfect to the cliff. You don't know. Some of us are born with certain genes right at the edge of the cliff. Anybody ever heard of the BRCA1 gene, BRCA1? Come on, guys, you've heard of that, hadn't you? The breast cancer gene, Angelina Jolie. Okay, that is a sequence. These people have inherited a sequence in that gene that is right next to the cliff. It can't accommodate many more changes until it falls off the cliff. Okay? Does that mean that those individuals that inherit that sequence are going to get cancer? No. It just means they don't have very far to go in mutations and accumulated mutations for that to happen. So normally people just monitor. People like Angelina Jolie go and have double mastectomies to totally eliminate the problem. Anybody in here live in Madison County? Now, Hudson Alpha, a biotech firm up there, is giving free genetic sequencing for the BRCA1 gene if you live in Madison County. I just offered that to anybody. I always try to tell people that because it's always good to know. Okay, so let's get all that in your notes. What's the origin of cancer? Well, those mutations in your DNA repair mechanisms. Also, mutations to proto-oncogenes. That's what the BRCA1 gene is. Proto means before. An oncogene is a gene sequence that results in those proteins not working and that those cells being cancerous. So proto means before a cancer-causing gene. So BRCA1 is a proto-oncogene. It can only accommodate a few more changes and it becomes an oncogene, which causes those individuals to have cancer. You also have molecules that, they're proteins, everything's a protein, molecules that suppress tumors in your body. Isn't that wonderful? You have molecules that suppress tumors, but they're proteins. So if you have mutation to those genes, you end up with cancer. When your cells divide, the end of every chromosome, okay, all 46 of them, the end is called a telomere. Tele means end. I don't know if you realize that yet. But the end of every chromosome is called a telomere. The end of the chromosome falls off during division. Oh, no. It can't do that, okay? It does. It falls off during division. And there is an enzyme called telomerase that builds it back. Well, telomerase is a protein. It's an enzyme. So if it's not working right, then those cells are allowed to divide continually. Cancer is over 100 different disorders. Okay, that's one of the reasons it's so hard to understand, because every type of cell is different. Remember, we said if we think of our chromosomes as a recipe book, every cell has every recipe. It's got the whole cookbook, okay? But not every cell uses every recipe. Different types of cells use different subsets of those recipes. Kidney cells would use different genes, different recipes than a liver cell. Colon cells would use an entirely different set of genes. So when a cell develops cancer, it's a different physiology. It's different in every type of cell. But they do have some things in common. So let's talk about what cancers have in common. Cancer cells lack differentiation. Cells start out as generic cells. You may have heard them called stem cells. Okay, when you were an embryo, you were stem cells. And then as the embryo develops, the cells differentiate. That means they take on an identity. They go from being a generic cell to being a kidney cell or being a liver cell. That's the normal progression. Once a cell differentiates, it doesn't go backwards normally. Okay? Cancer cells are cells that have differentiated. They were liver cells, but they've regressed and they have forgotten that they're liver cells. They don't have a job to do anymore. They are not differentiated. Cancer cells have abnormal nuclei. For some reason, they are misshapen, they're larger than they should be. They have abnormal nuclei. They also have the ability to form tumors. Most of you are familiar with what a tumor is. The reason they can form tumors is, well, let's back up for a moment and let's just say if you put normal cells in a petri dish and you give them lots of nutrients and room to grow, they will constantly divide, and that would represent growth, right? Once they fill up the petri dish and they come in really close contact with other cells, they have what's called contact inhibition, and they know to stop. They don't grow, they don't divide anymore. They only divide when another cell dies and needs to be replaced, and that would represent repair. Right? So normal cells divide for growth and repair. Cancer cells are not like that. They lack that contact inhibition. They continue to grow. They continue to divide even when they come in close contact with other cells. They have the ability to metastasize. You may have heard that word, that a cancer spread and metastasized to another location. That's a cancer cell breaking from one tumor, a primary tumor, and traveling to another location and forming another tumor. Just FYI, if someone has colon cancer and a cell breaks and goes to the liver and establishes another tumor in the liver, you can tell that's not a liver cell. Again, the physiologies are different. The things that cause those cancers to develop are different. So that patient medically would not have colon cancer and liver cancer. That patient would have colon cancer and colon cancer that metastasized to the liver. Okay? Because it's not a liver cell that has developed cancer. It was a colon cell. Okay? They have the ability to undergo angiogenesis. Angio means blood vessel. Genesis is beginning. So they literally have the ability to command your body to bring me nutrients. You have to have blood vessels to supply nutrients and oxygen to a cell. You have to have blood vessels to remove waste products from a cell. And normally blood vessels do not develop after an individual is matured. Okay, after development, new blood vessels just don't develop. Formation of new blood vessels. Anyone in here ever read the book, The Immortal Life of Henrietta Lacks? Okay. What you're seeing here, these are HeLa cells. They're called HeLa cells because of Henrietta Lacks. They were taken from her body. Back in the early 1900s, common practice was to take samples of people's tumors and to do research on. And primary research was to try to find cells that would live for a long period of time. Let me explain. When a cell divides and produces two cells, and those cells divide and produce more cells, and those cells divide, that whole line of cells, the original cell and every cell that came from that cell, is called the cell line. Okay? Cell lines don't last forever. They eventually will die. A cell can't divide eternally. Eventually, that cell line will die. Okay? The whole cell line will die. Well, researchers have been constantly trying to find cells that would live for a long period of time, division after division after division, because that's very important in research. If you're testing something, let's say you're testing a new drug, you want to make sure that you're testing the drug on cells that are exactly identical, because if they're not identical, you won't know if the results are from your drug or the differences in the cells. Okay? So researchers were constantly trying to find cells that would live for a long period of time. And here comes this lady, Henrietta Lacks. And she was a very poor woman in New England in the early 1900s. She developed cervical cancer. And as was the custom at that time, even without her permission or her knowledge, because there was no laws for forbidding it, the doctors took samples of her cancer. And they did it to every cancer. And they always named them. They used the first two letters of your first name, so she was Henrietta, so it would be H-E, and they used the first two letters of your last name. Her last name was Lacks, so it would be L-A, so the cell line was called HeLa, H-E-L-A cells, okay? Well, when they got the cells in culture, they started growing them, they realized there was something really, really strange about her cells. Her cancer cells. And they don't know if it's the cancer or they don't know if it's her cells, but something's very different about them. Her cells never die. They took that tiny, tiny sample of her cells and they started mass producing them. They never die. And we don't know why. To this day, we don't know why HeLa cells don't die. But her cells became the standard for all biological research. Anybody that's doing research orders HeLa cells to do the testing on. Her cells, her tiny, tiny sample of cervical cancer cells, are now a multi-trillion-dollar international business. Every drug that's been developed since the early 1900s, every substance that's been developed that has to be tested on humans has been tested on HeLa cells. I can't tell you how many tons of her cells have been produced to date, and they're still being produced because they don't die. Okay? It's just her cells from that tiny sample of cancer cells. Yes. But it's you can order them. I've thought about trying to order them just to show classes. These are HeLa cells because it is the standard. I mean, any scientist that's doing research orders HeLa cells. Okay, they're very expensive though. I decided that might not be a good budget request. Anyway, the sad part about this whole thing. I love the book. If you've never read the book, you really need to read it. It's very easy reading. It takes genetics and cancer and puts it on a level that anybody can understand. Okay? It's a very sad story. It's a very good book. The saddest part to me, though, y'all aren't going to believe this, the saddest part is the Henrietta Lacks family got not a penny from any of it. How sad is that? Yeah. Anyway, that's it for today. We'll have a quiz over this.